In this video, we are going to discuss about splicing. Before discussing splicing, we will look to certain terms. Spacer DNA. These are non-coding DNA present between the genes. These are found between the genes for 18 srRNA, 5.8 rRNA and 28 srRNA. Thus, the transcription of these genes will result into 45 as polycistronic rRNA. Polycistronic, these mini polypeptide chains will be formed from these mini chains. Mainly polycistronic is seen in the prokaryotes, but in eukaryotes, monocistronic, that is only a single polypeptide chain is formed from one single gene. Now, this spacer DNA is cut by enzymes called endonuclease. These endonucleases are the enzymes which cut the DNA at certain position between the sequences. This endonuclease cut away the spacer RNA and release the 3 rRNA. You can see in this diagram, this DNA is present and these are 18 sRNA, 5S and 20S RNA, and between these spacer RNA are present as ITS1 and ITS2. ITS1 is present between 18S and 5S and ITS2 is present between 5S and 28S. This ITS stands for internal transcribe spacer and ITS2. Similarly, ETS stands for external transcribe spacer. The transcription of these genes will result into 3 mature rRNA, 18S, 5.8S and 28S. Now, we will look introns. Introns, these are non-coding DNA present within the genes. Spacer DNA were present between the genes, but introns are present within the genes. And this separate the genes into coding and non-coding sequences. These non-coding regions are introns and the coding regions are exons. So RNA polymerase, it is an enzyme which transcribes the DNA into RNA. RNA polymerase does not distinguish between intron and exon. Both intron and exon get transcribed into RNA transcript containing both introns and exons, spliceosomes. Now, to remove the introns, the non-coding sequence from the primary transcript or the pre-transcript from the DNA, we require certain enzymes. These are spliceosomes. Now, how these spliceosomes work and what does it constitute, we will see it below. This spliceosomes is made up of RNA and protein complex. It mainly catalyzes the removal of introns from the nuclear pre-mRNA. This pre-mRNA is also known as primary transcript or the first RNA formed from the DNA within the nucleus. These spliceosomes will contain ribonucleoprotein complex. It is present in the nucleus and mainly found in the eukaryotic cells. See in this diagram, this RNA transcript or the pmRNA, it has both introns and exons. And what does the spliceosomes does? It makes a complex with protein and snRNA and other protein. Spliceosomes forms a complex where this RNA transcript uh, RNA transcript these spliceosomes form a complex where the RNA transcript get trapped. The exons is present in the upper side and the intron in the lower side. This intron will be cut and will form a loop and will be removed from these spliceosomes, and both these exons will be joined, giving us mature RNA. Splicing was discovered in 1970s. For most eukaryotic genes, the primary RNA transcript must be processed before it becomes mature mRNA. Now, this primary RNA As we have seen, it contains both introns and exons, and it cannot code for protein. Thus, it becomes mature mRNA by spliceosomes, and this mRNA, this mature mRNA, can directly synthesize protein. The RNA splicing involves removal of introns, ligation of the remaining sequences, that is coding sequences exons. It is carried out by enzymes spliceosomes in nucleus. Now, the spliceosomes, it consists of many different particles or substances in this four different sRNA. This snRNA stands for small nuclear ribonucleic acid, and snRNP, that is a small nuclear ribonucleoprotein. It forms a complex as both snRNA and snRNP works to bring the end of the exons in the primary transcript near each other. This snRNP is constructed from 6 to 10 proteins, one or two of the five snRNAs. These snRNA are named as U1, U2, U4, U5, and U6. These snRNPs are designated by snRNAs they contain. Introns are removed from primary transcript by cleavage at the conserved sequence called splice sites. So these introns are cut at the sequences which are called splice sites, which are found at 5' and 3' end of the intron. This RNA sequence that is removed or cut, it will begin from the dinucleotide GU at its 5' and ends with AG at the 3' end. The branch point, it is located 18 to 40 nucleotide upstream from the 3' end of the intron. So this branch point will have certain sequences. It is YN, YY, RAY, and it will contain adenine. This Y stands for pyrimidine, N for nucleotide, R for purine, and A for adenine.